Welcome to ASM Live, the internet talk show live from the ASM General Meeting 2013 in Denver, Colorado. We're here today to talk about a number of interesting aspects of microbiology, and I'm your host today. I'm Stanley Malloy. I'm the Dean of the College of Sciences at San Diego State University, and I'm the chair of the editorial board of Microbe magazine. I encourage you today, if you have any questions in the audience, to please come up to the microphone to ask questions. And if you're watching us online, please send any questions that you have to #ASMLive. With me this morning is Yuvon Mobley, and she works on antibiotic compounds isolated from wasp venoms. Could you tell us a little bit about what you've done, Yuvon?
Yes, so my research focuses on the use of these antimicrobial peptides against bacterial pathogens, and thus far we've been able to determine that they're highly effective against a broad range of bacterial pathogens, both gram-negative and gram-positive, and we've also utilized it in a bacterial skin infection model to show that it's effective against that as well.
So this is really interesting because peptides are these nasty compounds made by many, many different organisms, including part of our immune system. But there haven't been very many cases where they've really proven so far to be functional antibiotics. Is that right?
Yes, that is true. These peptides are produced throughout all different life forms in nature. As humans, we produce them in our skin and in our mucosal areas as well. And there have been some that are produced by insects and bacteria. However, a lot of times when we look at them as potential therapeutics, they're easily degraded in the body. A lot of them are very small, so proteases degrade them very frequently. And as well, they're typically highly charged, and so they also get caught up and kind of absorbed by some of the different cells in the body as well.
So how did you choose wasp venom?
So there was actually another group of researchers that had already identified this peptide from the wasp venom due to its ability to activate mast cells. And so that's how my lab became interested in it, because we're a mast cell and urinary tract infection lab. And so we were using this to activate mast cells, and then also decided to look at it for its antibacterial properties as well.
And do you think that this particular peptide overcomes some of the problems with the other peptides in terms of being stable inside the host?
Yes. So we've done some research, especially with one of our collaborators, Dr. Herman Statz, in looking at kind of the stability of the peptides, and also are looking at its potential as an adjuvant for vaccines. And so we really have seen that this peptide is able to kind of overcome some of those initial problems that they've seen with other peptides. And as well, we see that this peptide is even further advanced because it does both the antibacterial as well as the immune stimulatory properties.
That's very interesting. An adjuvant and an antimicrobial together in one package. Right? So you could have a vaccine that would be an antibiotic at the same time?
At the same time, yes.
Now, you've applied this so far in a model of skin infection.
Yes.
And those results are promising?
Yes, they are. So we've done both a colonization model where, I mean, as humans, bacteria kind of are part of our normal flora on our skin. And so we've done it where we've applied bacteria to the skin of mice and then allowed that to set up a colonization for about an hour. Then I've applied the Mastoparan on top of that, and we've shown that in both our log phase and our stationary phase bacteria, we've been able to reduce the growth of the bacteria in that way. And one of the things that's very surprising about that is a lot of our current antibiotics are not effective against stationary phase bacteria. However, with this compound, we are able to affect that stationary phase bacteria. And then we've also done a skin infection model where I go in, I do an intradermal injection with the bacteria Staph aureus, and that sets up an infection there as well. And we've treated the bacteria in those infections with our peptide, and have seen some very promising results with that.
So is that just a topical treatment with the peptide?
Yes, topical treatment.
And in the process of applying the peptide before you've applied the Staph aureus, does it disrupt the normal flora on the skin as well?
So we actually normally apply it after we've applied the Staph aureus. And in the mouse model, I think especially since when we shave and nair the mice, there isn't a large colonization that are naturally there on the mouse skin. But I think that once we go into the human aspect where there is that natural kind of flora that's there, there would potentially be some type of effect that you would see with that.
And in terms of how effective it is in this topical model, is it better, the same? How's it compared to other types of topical antibiotics that are used now?
Yes. So one of the major topical antibiotics that we use for comparison is Neosporin. And so there are 3 active ingredients in Neosporin that are antibacterials. And so we've compared it in comparison to that, and it has a 98% reduction greater than what we see with the Neosporin-treated mice.
That's very promising.
Yeah, so very promising.
Right. Now, a lot of these kinds of organisms that you would love to be able to treat because they're resistant to other antibiotics, many times they don't just cause a topical infection, they'll cause a systemic infection. Do you have any idea about how effective this approach would be in a systemic infection?
So we haven't technically looked at it in terms of systemic infection with our bacteria, but like I said, one of our collaborators has looked at it for adjuvant activity, and when we've applied it in the airway and in the nose, we have seen kind of increases in IgG and IgE more systemically with that type of application. So I would kind of branch that out and say that most likely we would be able to see something and some type of effect with this more systemically, even against bacterial pathogens.
And does it stay in the bloodstream for a reasonable period of time?
That's not something that we've looked at thus far.
Well, I think that that's really interesting and promising. Not only for this peptide, which is really exciting, but I think that what you learn with this peptide, how it might make us more aware of other types of peptides, or even of modifications of the peptide. Have you thought about aspects like that?
Yes, so actually there are 2 other homologs of Mastoparan, Mastoparan 7 and Mastoparan 17, that have 2 amino acid substitutions, and that has made Mastoparan 7 a more potent mast activator and Mastoparan 17 inactive. And so we've looked at some of those substitutions and made some substitutions at the similar locations, and what we've actually found is that in terms of its mast cell activation, we've gotten increased mast cell activation with these substitutions. However, with our antibacterial looking at that way with it showing its antibacterial properties, we haven't seen any additional effects with that. And so I really think that evolutionarily, that with this peptide being in the wasp venom, somehow has maximized its antibacterial properties with the current peptide structure.
So again, I encourage anyone in the audience to ask questions, and we're taking questions on Twitter, hashtag ASM Live, if you want to add to the conversation today. So one of the really important aspects of an effective broad-spectrum antimicrobial is that it has to hit gram-positive bacteria like Staph aureus, which you mentioned, but also gram-negative bacteria, and sometimes membrane-active agents, like this compound, don't work as well on gram-negative bacteria. Is that the case for this compound?
That is not. So we've actually tested it against both our gram-positive and our gram-negatives, and it has similar activity and potency against both of those 2 types of bacteria, which is important to note because a lot of our current antibiotics are typically only effective against either the gram-negatives or the gram-positives, whereas with this compound, we have both of those activities combined in one peptide.
And so the mechanism of action, it interacts with the membrane and causes lysis, is that?
Yes, exactly. So we've done some microscopy with this and shown that there is some membrane interaction that happens, and we can even see with some of our scanning electron microscopy, where you see beautifully where you can determine exactly where the peptide has interacted with that membrane and caused kind of these pores or holes in the membrane of the bacteria.
Formed really quick?
Yes, within 30 minutes.
Now, does it have any impact on eukaryotic cells?
So we've done some tests on eukaryotic cells, especially in trying to determine whether or not it would be safe as our topical model, and we have shown that, at extremely high concentrations, and yes, there is some interaction with our eukaryotic cells, but at the concentrations that we've seen for our antibacterial activity, there's nothing that happens with our eukaryotic cells.
That's very interesting for an agent that interacts with the membrane, right? So you say, what's special about the bacterial membranes? It's different in the eukaryotic membranes. It's resulting in this difference. Do you have any clues about what that might be?
We don't know exactly, but we think a lot of it has to do with the protein content that makes up the eukaryotic cells, as well as the difference in the charges between our eukaryotic cells and the bacterial cells. Because this is a highly charged peptide, and so we think that a lot of the interaction kind of goes along with the charge. And as well, the peptide is amphipathic, which means there's kind of charged regions on the end and non-charged regions in the middle. So that allows it to associate with the hydrophobic and hydrophilic regions of the bacterial membrane to cause those pores and those holes.
Now, for something like this to be effective, you need to produce a lot of it.
Yes.
And is it easy to produce?
Yes, at this point we've determined, I mean, we know the 14 amino acid sequence of it, so a lot of times we can just go to a company and have them kind of come up with that sequence. We're no longer going out and catching the wasp and kind of extracting it that way at this point. It's just being produced by a company.
Did you ever have to do that?
I didn't, thankfully.
There's some people in the world who really hate wasps that
Yes.
would probably be happy to see you decimate them, but I think maybe the plants wouldn't like that as much.
Right.
So you can produce it on pretty high quantities
Yes.
then and test it. And what kind of concentrations are required for it to be effective?
So when we're looking at our antibacterial concentrations in vitro, I'm typically around about a 7.8 to a 62.5 micrograms per mil concentration. For our in vivo studies, we've been using a 10 microgram concentration.
Mm-hmm. And do you know if resistance develops to these compounds?
So we've done some studies in terms of resistance development. We've used a high mutator strain of bacteria, and we've been applying dosages of Mastoparan every day to that concentration of bacteria to see if there is any type of resistance development. And thus far in our studies, we've shown that there isn't. And it takes a longer period of time for us to see resistance development to this compound than what we see to some of our conventional antibiotics.
That's very interesting because there's as we started off talking about, there's so many of these peptides out in nature,
Right.
antimicrobial peptides, and so it's one of these situations you would think that the host would modify and then the bacteria would modify and the host would modify, and so you'd have this constant evolution of resistance and sensitivity. But
I think one of the major things of note is the mechanism of action. And since it is having that cell membrane interaction, it really is somewhat difficult for bacteria to kind of change their cell membrane in order to evolve some type of resistance mechanism. I think that with a lot of our other antibiotics where they're going in there inhibiting translation or transcription, it's a little bit easier for them to change kind of the conformation of those proteins to be able to avoid and become resistant to those, versus with the cell membrane, it's a little more difficult.
Mm-hmm. So one of the other really important questions when you're thinking about antimicrobials is mechanism of delivery. Right? Now, in the case of topical, that's about as easy as you can get.
Yes.
But what would be really nice would be if you could deliver orally. Do you think that there's hope of delivering peptides like this orally?
I mean, it's always possible. I think that with these peptides, especially since they are pretty small and easily degraded, orally may not be the route to go, but I do think that something that's more of a topical type of antimicrobial situation would probably be the best route for this.
And what do you imagine in the future, given where we are right now with the level of multidrug resistance that really is a serious problem? How do you imagine the antimicrobial peptides will affect us 5 years down the road or 10 years down the road? Clinical trials take a long time.
Yes. Well, I think that, like you said, the increase in antibiotic resistance that we're seeing in these bacterial pathogens is becoming a global health problem, and that we really are in dire need of some new strategies, and really especially new strategies that utilize a unique mechanism of action, such as these peptides, to kind of help us to combat these types of infections. I think one of the good things with some of these peptides, such as the one that I work with, Mastoparan, is the fact that it has both that antibacterial activity as well as the ability to stimulate the natural and innate immune system, which really not only kills the bacteria directly, but also boosts up your natural immune system to help you clear that infection as well. And I think that this is really kind of the way to go. And there's a lot of research where we're looking at kind of the two between antibacterial and immune stimulatory molecules, because that combined action really will give us an even greater clearance of the bacteria.
That's great. We have a question now from the audience.
I was just wondering, is this going to be useful?
And so there are several different components of wasp venom that would help in terms of causing the allergic response. So this is one small peptide that's combined in that doesn't typically have that. I know a lot of people are also pretty concerned about, oh, well, if you're activating mast cells, then would that also cause some type of adverse reaction or anything like that? In terms of my studies, we have gone and applied this peptide in very high concentrations onto the skin of the mice and have also injected it underneath the skin of the mice for several dosages over a 2-week span of time. We haven't seen any inflammation or anything like that at the site of injection or at the site of application. However, there would potentially be a concern if there is someone that does have or is highly allergic to certain things that they may have some type of reaction. I think that's with a lot of the current antibiotics and compounds that are out there. Depending on what you're allergic to, you may have an adverse reaction to it.
So the question, I guess part of the question too is, are there potentially compounds within the wasp venom that would prevent the allergy instead of accentuate the allergy?
So I'm not certain. Like I said, this is only one small component to that wasp venom. There are other things contained in the wasp venom. This was just a component that they identified that had the ability to activate mast cells. So there are other things, and I think even if we think about why the wasp would have a peptide like this, there's been some current research in looking at why, from the wasp aspect, there would be this type of peptide. And really, wasps were parasitoid, which means that they inject their venom along with their eggs into things such as cockroaches and flies. And as we know, cockroaches and flies come into contact with a large variety of microorganisms throughout their lifetime. So it's thought that these wasps have evolutionarily kind of obtained these types of peptides within their venom to help protect their eggs and the growing larvae from the different microorganisms that they may come into contact with once they've been injected into the body of a host such as a cockroach or a fly.
I was just going to ask you about the wasp perspective.
Yes.
We think so much about humans, we often forget that this is out in
Right.
nature. But along those lines, then, if this is in the wasp venom, is it also in a high concentration in the larva?
So several different wasps kind of utilize different mechanisms. There are some larvae that themselves produce similar peptides, as well as it being encompassed in the venom itself. So it's not in a very high concentration. I know we haven't really done, and there haven't been a ton of studies from the wasp aspect. So I don't know exactly what concentration it is in the wasp, but I would expect it to be something similar to what we're seeing, since it is there to kind of help minimize the type of microbial that the larva would come into contact with.
So that argument helps you understand why it might be antimicrobial. What about the inflammatory aspects? How do you explain that if you're a wasp?
And so a lot of that in terms of like the mast cell activation. So if a wasp kind of stings a human, which isn't normally what the wasp would sting, but if a wasp does sting something like a human, those mast cells kind of help with vasodilation of your blood cells so that the venom would be able to circulate more freely and quicker through that body.
Makes sense. And of different types of wasp, is this particular peptide common in many different wasps?
Yes. So Mastoparan is actually a family of peptides that we see throughout different types of wasps. The one that I specifically work with does have a specific 14 amino acid strain, but there are other peptides similar to this that are found throughout wasps. They're also found in the skin of frogs. Scorpions also have similar types of peptides as well in their venom.
So you imagine that you would have a whole repertoire of different peptides.
Yes.
And as resistance develops, you could just move on down the line.
Perhaps, yes, you could try out one, and if that one doesn't work, or once resistance develops, you can go to a different one.
In contrast to antibiotics, where in fact we have relatively few available antibiotics to
Right,
use.
exactly.
How do you imagine that, well, is there any interest from industry in your product so far?
We haven't seen any interest from industry thus far. I mean, I definitely think that what we have and kind of Mastoparan in general is a very, very promising therapeutic. So hopefully we'll be able to drum up some support from industry for this.
Why do you think that they're not already excited? Do you think it's because of concerns about clinical trials or other studies on peptides? Or you have a clue about that?
I think part of it is that we haven't, I mean, this kind of is the first meeting that I've gone and presented about my data. So it hasn't, the information hasn't really readily been out there thus far. I think that once we do start getting the information out there and once a publication comes out about it, that will hopefully drum up some additional support from industry.
What do you imagine are the next steps in the research process to begin to move this forward?
So I know part of it would be to actually go into researching whether or not we can go into clinical trials with this. And another thing that we've looked at in our lab is trying to see if we can develop similar peptides such as this and this peptide into a nanoparticle formulation to see if we can kind of enhance the delivery mechanism of it to kind of help with some of the degradation that you see by proteases and that type of thing, and help, once you've applied it, for it to have a more sustained and prolonged type of activity via nanoparticle formulation.
Yeah, that's very exciting. It takes in the synthetic and the natural, mixing them together in a very effective way.
Right.
If there were people out there thinking of this as a potential research topic, or students thinking about what they're going to do with their careers in the future, what do you think your story tells them about opportunities in microbiology and science?
I mean, I think it's very exciting. I know that specifically from my research, I think being able to see kind of more of the therapeutic aspects and seeing this as something that really can have kind of broader global applications really is promising and exciting to know that the work that I'm doing in the lab really could someday affect millions and billions of people and affect their lives and really try and help the society as a whole.
You know, one of the interesting things about these peptides versus antibiotics per se is that in nature there's a high abundance of antibiotics out there, and nobody really knows why. There are a lot of theories out there, one of the theories being that they're involved in signaling. But in the case of these peptides, it really looks like they are there for this specific purpose, right?
Yes.
It makes you rethink the idea of natural resistance and how you develop natural resistance and the long-term effectiveness of these as a product if you can overcome that initial hurdle.
Right. Yes, like I said, I really think that especially in some of our tests where we've tried to kind of modify the structure of the natural peptide and have seen that in terms of its antibacterial properties that we lose that effectiveness, that nature really has worked to maximize that structure and has really kind of evolutionarily conserved that structure there to make sure that it's kind of the best that it could be.
Do the inflammatory properties of these peptides also influence virus infection?
So we haven't technically, I know there are a couple of other labs that have looked at it with virus infections, but we haven't done any virus infection work with it.
Do you know if people have looked at parasites as well?
Not that I know of. Not specifically with this peptide. There are other antimicrobial peptides that individuals have looked at in terms of its effectivity against parasites.
How long do you think it might be before something like this would make it to market?
I mean, I'm hoping, especially with this growing antibiotic resistance that we're seeing and the superbugs and the increase that we're having with that within the next 5 to 10 years that we really would see something like this on the market.
5 to 10 years is a very short time frame.
Yes, it is. But like I said, the growing need that we have for kind of clinical antibiotics with such as this really makes it hopefully at the top of the list of things that we should be exploring.
So if it was 5 to 10 years, that means you're going to want to begin to think about clinical trials very, very soon. Yep. Are the plans already in the works?
Not from our end. Like I said, hopefully once we've kind of drummed up some initial support and gotten some reactions about this research, that someone will take it and run with it.
You going to start your own company?
That's not currently in the plans, but I mean, if someone came to me with a good offer, I don't think I'd turn it down.
So anyone out there on the Twitter feed of hashtag ASM Live with a question or an offer to support funding of this company, why, let us know.
Definitely, let us know.
Now, you've been doing this for your graduate education. Yes. Where are you going after you finish graduate school?
So yes, most of my graduate research has been focused on trying to identify how well this peptide works and if we can utilize it as a potential therapeutic. Next steps for me will be looking for a postdoc. I'm not certain where yet, but once again, if anyone out there in the audience or in our Twitter world has an opportunity for me, I'd love to talk with you.
Are you planning to continue working in the antimicrobial peptide sphere?
Somewhat. I'm not necessarily tied to it, at least not now, unless I get an offer from, like you said, a company to start up something. I'm really open to any area of research.
Well, I think this is particularly interesting because of the potential to think about nanoparticles and other synthetic approaches coupled with peptides that have for a long time been problematic because of stability, because of difficulty of delivering to target, and so on. So it really is an exciting time for science, for these types of discoveries to have a real impact on people's lives. Right? And I think that there's one other thing that's really interesting about this, and that is, you could imagine the first person that submits a proposal on antimicrobial peptides from wasps, and someone in Congress saying, what the heck is this? Why are we funding research on wasps, right? What do we care about wasps? Whereas, in fact, it's an example of one of those kinds of discoveries that has the potential to profoundly impact human health. So,
I agree. So I think that sometimes, especially as students, we may start working on something, we may think, oh, well, this isn't anything that's that interesting or doesn't have that broad spectrum of application, but you never know. You can be surprised by what it is that you can discover.
Well, it's been very nice talking to you.
Yes.
Thank you so much.
Thank you.
I look forward to seeing these products make it to market and save people's lives.
Exactly. Thank you.
Thank you.
Extending the legacy of a classic reference, EcoSal Plus joins the ASM family of periodicals this June. This growing collection of continually published reviews reveals our ever-expanding understanding of the biology of E. coli, Salmonella, and the other Enterobacteriaceae. From cell architecture and growth to pathogenesis of infections to life in communities, EcoSal Plus is the single source of knowledge about these organisms and their use as model microbes. A distinguished editorial board, led by Editor-in-Chief James Caper, ensures that content serves the needs of the research community. Free trials available for institutions. Take a look at ecosalplus.asmblog.org.